This method combines the ability to stain for a membrane protein while also preserving membrane integrity and subcellular architecture. So it's really powerful for precisely localizing a membrane-associated protein.
The main advantage of this technique is that it combines the use of a robust genetically clonable tag, APEX-2, with state-of-the-art methods in cellular preservation for electron microscopy, including cryofixation and freeze substitution. Together, these allow precise protein localization within a well-preserved cell.
This protocol can be used to dissect mechanisms by which viruses replicate inside the host cell. Viruses often will hijack host proteins during their infection scheme. Viruses harbor small genomes which they utilize to replicate inside these host cells. Recently, we've been able to, in unpublished data, label these proteins using this technology to understand how viruses replicate.
This technique proved really critical in a recent collaborative study on a bacterial toxin that causes Golgi breakdown. Using CryoAPEX, we were able to precisely localize the toxin with the fragmented Golgi pieces, something that was really difficult to see otherwise using normal immunofluorescence techniques. Demonstrating this technique will be Elaine Mihelc, a graduate student, and research associate Stephanie Angel from our laboratories.
After seeding HEK293 T cells in a dish, transfect the cells with APEX-2 tagged mammalian expression plasmids using transfection reagent according to the manufacturer's directions. At 12 to 15 hours post-transfection, wash the cells once with PBS. Then, wash off the cells with PBS.